Welcome to the pod Biomanufacturing and Fermentation Technology. In this episode, we translate microbial science, process engineering, and manufacturing realities into insights that scale.
For decades, industrial biomanufacturing has operated on this kind of dirty little secret.
Oh, absolutely. It's a massive open secret in the field.
Right. Like, to keep engineered cells producing at scale, you basically have to pump your fermentation tanks full of expensive antibiotics.
Yeah, it's the only reliable way to maintain genetic stability.
And if you are a scientist or, an industry expert managing downstream purification, this unspoken, frustrating reality intimately.
It is arguably one of the most stubborn bottlenecks we face in industrial biotech today.
But May 2026 paper from Nature Chemical Engineering just blew that paradigm apart.
It really did.
Yeah.
We are finally seeing a structural decoupling of genetic stability from that, chemical enforcement.
Yeah, and this month, the laboratory promise of antibiotic-dependent high-titer fermentation was shattered at pilot scale.
In a commercially relevant process, no less. It's huge.
Okay, let's unpack this, because our mission for you today on this deep dive is to completely deconstruct how these researchers achieved this.
Right, something that has historically been basically an oxymoron.
Exactly. High-titer, antibiotic-free, pilot-scale production of 1-glur-3-propanediol or 1,7,3-PDO.
Which is a massive platform chemical, by the way.
So we are going to examine the metabolic rewiring, the actual physics of the scale-up, and the hard downstream economics.
But to really get this breakthrough at the 300-liter pilot scale, we have to start at the foundation.
Right, the foundational metabolic engineering that kicked the research off. Where did the genetic architecture actually begin?
So the foundational work started with Corynebacterium glutamicum, ATCC 13302.
That's the standard one.
Right, yeah, exactly. In the realm of systems metabolic engineering, this strain is, well, it's the classic laboratory sandbox.
Because it's just so easy to work with.
Precisely. It is highly tractable, the genome is completely mapped, and the molecular tools to manipulate it are incredibly well developed.
So the researchers utilized this strain to, I guess, establish the baseline genetic architecture.
Right. They needed to force the organism to consume glycerol and synthesize 1,3,3-PDO.
But naturally, Corynebacterium isn't optimized to just churn out industrial quantities of a solvent-like molecule from glycerol.
No, not at all. It doesn't want to do that.
So you have to actively dictate the intracellular carbon routing.
You do. They had to introduce heterologous glycerol uptake pathways and the specific 1,3-PDO biosynthetic enzymes.
Just putting the machinery in there.
Right. But in metabolic engineering, introducing a pathway is really only half the battle.
What's the other half?
The rest is about managing the thermodynamic flow of carbon. To do that, they performed a series of targeted gene deletions.
To close off competing metabolic branches, I assume?
Exactly. Instead of thinking of it like a highway with off-ramps, think of it as closing thermodynamic escape valves.
Oh, I like that analogy.
Yeah, so by knocking out pathways that naturally lead to byproducts like lactic acid or acetic acid, you change the flow.
The intracellular carbon flux has this energetically favorable funnel directing it straight toward 1,0,3-PDO.
Precisely. You give the carbon nowhere else to go.
I don't get something here, though. The methodology outlines all of this highly complex, elegant genetic rewiring.
Yeah, it's very thorough work.
Like mapping the fluxes, validating the deletions, all within that ATCC 13032 strain, but then they execute this massive pivot.
Right. They switch the chassis entirely.
Yeah. They take this entire optimized genetic architecture and transfer it over to a completely newly isolated strain, C. glutamicum SC97.
Which was a brilliant move, frankly.
But why go through the trouble of porting this entire architecture over to the SC97 strain instead of just brute forcing the original ATCC 13032?
I mean, it sounds crazy on paper.
It seems incredibly risky and labor-intensive to do that at the 11th hour.
It is, but that pivot gets to the absolute heart of what we call chassis robustness.
Okay, break that down for us.
Well, you can design a flawless metabolic pathway on a computer, right?
Yeah.
And you can achieve perfect redox balance in a 1-liter flask.
Sure, where conditions are perfect.
Exactly. But if the underlying organism, the chassis itself, cannot handle the baseline physiological stress of industrial production, it just fails.
The entire system collapses during scale-up.
Right. The ATCC 13032 strain is a phenomenal sandbox for prototyping genetics, but it just lacks the native resilience for heavy manufacturing.
So the actual cellular machinery just buckles under the metabolic burden.
Inherently, yes. When you're forcing an organism to accumulate massive intracellular titers of an unnatural chemical like 1,3-PDO, it's rough.
Because it's basically toxic to the cell, right?
Exactly. You are subjecting it to severe osmotic stress and solvent toxicity.
And the SC97 strain is different.
Very. The SC97 strain is a newly isolated wild-type variant. It possesses a naturally robust membrane integrity and a highly active stress response network.
So it can just take a beating.
Exactly. The researchers recognize that an optimized metabolic pathway is completely useless if the cell's core energy metabolism is overwhelmed.
Just trying to survive the toxicity of its own product.
Right. Transferring the genetics to the SC97 chassis wasn't a detour. It was a fundamental prerequisite for surviving the harsh realities of the pilot scale.
Okay, that makes total sense. So if you have this incredibly robust SC97 strain carrying all these heavy genetic modifications, there's a new problem.
Maintaining the plasmid.
Right. How do you actually keep it from just ditching the engineered plasmid to save energy, especially if we are taking away the antibiotics?
That is the million-dollar question in biomanufacturing right now.
Because historically, you attach an antibiotic resistance gene to your production plasmid, right?
Yeah, and you maintain a high concentration of antibiotics in the fermentation broth.
So if a cell drops the plasmid, it loses the resistance gene.
And the antibiotic in the broth kills it. It's a brute force selection method.
But to circumvent this, the researchers engineered what is known biologically as a toxin-antitoxin, or TA module, directly into the plasmid.
Which is just a brilliant use of synthetic biology.
Walk us through the biological mechanism of that TA module. How does a cell actually police its own genetics?
It relies on differential protein degradation.
Okay, what does that mean in practice?
Well, the engineered plasmid constantly expresses two things. First, a highly stable, long-lived cellular toxin.
Sounds dangerous.
It is. And second, an unstable, short-lived antitoxin that binds to and neutralizes that toxin.
So as long as it has the plasmid, it has the antidote.
Exactly. As long as the cell retains the plasmid, it continuously produces both. And the antitoxin keeps the cell perfectly safe.
But if the cell attempts to shed the metabolic burden by dropping the plasmid,
the production of both proteins stops. And because the antitoxin is highly unstable, it is rapidly degraded by cellular proteases.
Oh, wow. But the toxin sticks around.
Right. The long-lived toxin remains active in the cytoplasm. Without the antitoxin to neutralize it, the toxin attacks the cell's own essential processes.
And the cell dies.
Instantly. It's remarkably effective.
Here's where it gets really interesting. You're comparing traditional antibiotic selection to an external enforcement squad.
Right, one that requires constant, expensive chemical feeding to keep the cells in line.
Exactly. But plasmid addiction is an internal dead man's switch,
where the cell polices its own genetic stability intrinsically.
If the cell abandons the factory blueprint, it triggers its own destruction.
What's fascinating here is the sheer biochemical elegance of that dead man's switch when applied to industrial scale.
Because it frees up resources, right?
Exactly. By removing the external antibiotic pressure, you drastically reduce the overall metabolic load on the culture.
The cells are no longer forced to waste precious intracellular ATP synthesizing massive quantities of resistance proteins.
Like beta-lactamase, yeah. They don't have to do that just to survive the broth. That preserved energy can be redirected.
And they didn't just secure the plasmid with that dead man switch, they combined it with another specific genetic modification.
Right, the overexpression of SucceD.
What does SucceD actually do in this context?
So the SucceD genes code for an enzyme complex called succinyl-CoA synthetase.
Which is part of the Krebs cycle, right?
Exactly. It operates deep within the cell's core metabolic engine, the TCA cycle.
And overexpressing it does what?
By overexpressing SucceD, the researchers fundamentally altered the intracellular kinetics.
Like speeding up the engine.
In highly simplified terms, overexpressing this specific node in the TCA cycle acts almost like a metabolic vacuum.
A vacuum. Okay, I like that.
It actively pulls the upstream carbon flux, so the raw glycerol entering the cell, down through the pathways at an accelerated rate.
Channeling it directly toward the 1,3-PDO biosynthesis route.
Precisely. It creates a thermodynamic pull.
So you secure the structural blueprints with the toxin-antitoxin dead man switch.
Right.
And then you supercharge the carbon flow with that SucceD vacuum effect. It is a brilliant two-pronged strategy.
It really is elegant metabolic engineering.
But if they've theoretically decoupled stability from the chemical environment using the system, does that actually translate to the real world?
You mean surviving the physical beating of a massive industrial tank?
Exactly, because the transition from the benchtop to the pilot plant is where so many projects just die.
Oh, completely. But that is where we see the true value of this integrated engineering.
Let's talk about the numbers. In their lab-scale fed-batch bioreactors, utilizing a mixture of glucose and glycerol, what did they get?
The culture yielded an impressive 141.5 grams per liter of 1,4V3 PDO
Wow, at what rate?
at a specific rate of 2.95 grams per liter per hour.
That's fast. And what about when they restricted the lab fermentation entirely to pure glycerol?
Eliminating the easy carbon source of glucose, they still reached 100.9 grams per liter.
Wait, really? Just on pure glycerol?
Yeah, the chassis is that efficient.
Those are phenomenal lab metrics, but the numbers from the scale-up are what actually dictate commercial viability.
Absolutely. The bench is easy compared to the pilot plant.
When they moved this out of the highly controlled 1-liter lab bench setup into 30-liter, and eventually 300-liter pilot fermentations, what happened?
They maintained yields of 120 to 128 grams per liter.
That is staggering.
And remember, they maintained that with absolutely zero antibiotic selection pressure.
For you, the listener, we need to emphasize how rare it is to see lab metrics hold steady in a 300-liter bioreactor.
It's almost unheard of without severe optimization.
Typically, when moving to those volumes, we see a massive cliff in what engineers call specific productivity, or QP.
All right, QP just tanks.
Why do typical industrial conditions usually destroy the QP of engineered strains?
It comes down to reactor hydrodynamics, basically the messy, unavoidable physics of scaling up fluids.
Okay, explain specific productivity first.
So specific productivity, or QP, is the measure of how many grams of your target product a single gram of cellular biomass can produce in 1 hour.
So it's a measure of per cell efficiency.
Exactly. In a 1-liter lab reactor, the mixing time is measured in milliseconds.
Everything is perfectly balanced.
Right, the environment is perfectly homogeneous. Temperature, dissolved oxygen, and nutrient distribution are entirely uniform.
Every single cell experiences optimal conditions simultaneously.
But you can't instantaneously mix 300 liters.
Let alone a 10,000-liter commercial tank.
Exactly. As the volume increases, the mixing time stretches from milliseconds to seconds, or even minutes.
So the environment fractures.
Yes. You must introduce massive impellers to agitate the fluid. This creates severe mechanical shear stress.
Literally physical trauma to the cell walls near the impeller blades.
Right. And simultaneously, because the mixing isn't instantaneous, chemical gradients form. You develop distinct microenvironments within the tank.
So a cell might be circulating near the top baffles, completely starved of oxygen and carbon.
Yeah.
And then seconds later, the fluid dynamics sweep it down directly into the feed zone where it is suddenly overwhelmed by a massive concentration of glycerol.
Exactly. Those oscillating microenvironments subject the cells to continuous, chaotic shocks.
And how do normal cells handle that?
Poorly. In standard laboratory strains, these rapid fluctuations trigger what is known as a stringent response.
The cells basically panic.
Yes. They rapidly divert their intracellular ATP and resources away from the specific productivity of your target chemical.
To save themselves.
Right, exactly. They redirect it into synthesizing chaperone proteins and repairing membrane damage. They shift from a production state to a strict survival state.
And that is the fundamental reason QP crashes during scale-up.
You nailed it.
So how did this specific integrated system, the SD97 strain coupled with the dead man switch, manage to survive the chaotic hydrodynamic environment without panicking?
Well, the robust SC97 chassis possesses the native physiological tolerance to absorb the physical shear stress and the osmotic shocks.
So it doesn't immediately trigger a stringent response.
Right. It doesn't waste energy overreacting to the chemical gradients.
Then what about the plasmid?
Concurrently, the toxin-antitoxin plasmid addiction physically prevents the cells from taking the easy way out.
Because if they drop the plasmid, they die anyway.
Exactly. Even under fluctuating stress, the internal genetic logic dictates that they cannot shed the heavy 1,7,3 PDO pathway to save themselves.
By combining the physical resilience of SC97 with the intrinsic genetic enforcement of the TA module, the system basically effectively decoupled genetic stability from environmental variability.
That is exactly what they did. The cells remained locked in a high production state, preserving that critical QP across the gradient stress of a 300-liter tank.
So what does this all mean?
It means we have a viable path forward for real-world production.
Right. The metabolic engineering is elegant. The sheer physics of maintaining QP at 300 liters is an engineering triumph.
He really is.
But if the unit economics fall flat, the industry will simply ignore it. Let's look at the techno-economic viability.
Which is what really matters for SMEs and founders.
Exactly. 1,3-PBO is heavily utilized as a platform chemical for high-performance polyesters, right?
Yes, but it is also a massive component in the cosmetics industry.
Which is a huge market.
It is highly prized in cosmetics as a pure, non-irritating solvent and a humectant. It replaces petroleum-derived glycols.
But the regulatory standards for cosmetics dictate a level of purity that makes traditional biomanufacturing economically punishing.
That's putting it mildly.
I mean, if you are manufacturing 1003 PDO using traditional antibiotic-dependent fermentation, your downstream processing, the DSP, is an absolute nightmare.
Oh, it's brutal. You cannot have residual kanamycin or beta-lactam resistance genes in a moisturizing cream.
Right, a cream that people are actively rubbing into their skin.
Exactly. The downstream processing in biotechnology frequently accounts for 50 to 80% of the total manufacturing cost.
Just separating the product out.
Yes. To guarantee the total absence of those antibiotics and resistance genes to regulatory agencies, you are forced to implement intense procedures.
Like chromatography and stuff?
Right. Layers upon layers of intensive chromatography, ultrafiltration, and complex separation steps.
So you are spending massive amounts of capital expenditure and operating expenses.
Just to remove the chemical enforcement squad that you were forced to pay for in the upstream bioreactor?
It's an insane paradox. But by utilizing the SC97 chassis with the intrinsic plasmid addiction, you are entirely eliminating the presence of antibiotics at the upstream fermentation stage.
Which is the game changer.
This fundamentally rewrites the cost structure of the downstream processing.
It does. You bypass those intense chromatography steps entirely.
Slashing both regulatory compliance costs and the massive raw material costs of the antibiotics themselves.
And if we connect this to the bigger picture, the techno-economics are further validated by the specific feedstock they used.
Oh, right. What did they feed this chassis?
They specifically modeled and executed this pilot process utilizing biodiesel-derived waste glycerol.
Waste glycerol, so they are actively avoiding the use of highly refined sugars.
Right, meaning this bioprocess doesn't compete with the global food supply.
That's a huge point for sustainability metrics.
Absolutely. The production of biodiesel generates millions of tons of crude glycerol as an unavoidable byproduct.
And historically, what happens to that?
Historically, this crude glycerol has been a low-value, highly impure waste stream. It is often a financial liability for biodiesel manufacturers to dispose of safely.
So they basically get paid, or at least save money, by taking it.
Yeah, by engineering this inherently tough Corynebacterium chassis to not just tolerate, but actively thrive on that specific crude waste stream.
Converting it efficiently into a premium platform chemical like 1,3-PDO?
Exactly. The researchers completely validated the industrial economics. Their life cycle analysis confirms it.
It is the absolute holy grail of the circular bioeconomy.
It really hits every mark.
You are taking a distressed industrial waste stream, feeding it into a massive bioreactor populated by a self-policing, highly robust organism.
And extracting a high-value chemical without ever introducing a single drop of antibiotics into the facility.
For the founders, bioprocess engineers, and SMEs analyzing this is the blueprint for how you enable valid, economically sound green claims.
Exactly. It isn't just marketing fluff.
The downstream capex is slashed, the raw feedstock costs are marginalized, and the environmental footprint is drastically reduced at a true commercial scale.
It perfectly exemplifies the necessity of integrated systems engineering.
You have to look at the whole picture.
Right. You cannot isolate the genetic design of a pathway from the hydrodynamic physics of a 300-liter tank.
Nor can you separate the upstream fermentation from the downstream purification costs.
The researchers succeeded here because they engineered the fundamental genetic logic of the cell with the final, harsh industrial reality in mind from day one.
Let's do a rapid-fire recap of the key technical mechanics we've deconstructed today.
Sounds good.
First, we explored the critical necessity of chassis robustness.
Right. We saw why porting the optimized genetic architecture away from the fragile ATCC 23032 lab strain was mandatory.
Moving it into the highly resilient wild-type SC97 strain was a prerequisite for surviving the osmotic and solvent stress of industrial titers.
Second, we examined the biology of replacing external antibiotic pressure with a toxin-antitoxin plasmid addiction module.
That intrinsic dead man switch.
Exactly. Functioning synergistically with the succeeding metabolic vacuum effect, it structurally guaranteed genetic stability.
And high thermodynamic flux without wasting cellular ATP on resistance proteins.
Third, the definitive proof was in the pilot scale physics.
The integrated system didn't just survive the scale-up.
It preserved a highly efficient specific productivity, QP, within the chaotic mixing, shear, and oscillating chemical gradients of a 300-liter bioreactor.
Maintaining yields of 120 to 128 grams per liter.
Finally, we contextualize the techno-economics.
By metabolizing crude waste glycerol from the biodiesel industry.
And by entirely bypassing the punishing costs of downstream antibiotic purification required for cosmetic-grade chemicals.
This research presents a commercially ready, highly profitable pathway for green biomanufacturing.
It is a phenomenal, paradigm-shifting piece of work out of nature chemical engineering.
But before we wrap up this deep dive, I want to leave you with a final thought to mull over, something that builds on the implications of what we've just discussed.
Where do we go from here?
Exactly. We have just examined how an intrinsic biological dead man switch can successfully replace externally applied antibiotics for genetic maintenance and a massive industrial tank.
Which is incredible on its own.
But if we can successfully offload that vital control mechanism directly into the cell's own genetic logic,
What else do we do?
what other traditional chemically enforced bioprocessing parameters could we eventually eliminate?
That's a profound question.
Could we engineer a chassis with internal logic gates so highly sophisticated that it requires absolutely zero external process control beyond the raw feed?
Just let the cells run the entire reactor.
Right. Imagine a 10,000-liter bioreactor where the physical addition of external bases for pH buffering is obsolete.
Or the manipulation of cooling jackets for temperature shock responses.
And the addition of chemical defoaming agents. What if all of those parameters were managed intrinsically by the cell's own engineered feedback loops and biological sensors?
That level of autonomous cellular engineering would fundamentally redefine the physical footprint and capital expenditure of industrial biomanufacturing globally.
At the start of this deep dive, we talked about how the long-standing, frustrating rule of antibiotic-dependent fermentation was finally shattered.
And it truly has been.
Well, as this SC97 Corynebacterium chassis has so clearly demonstrated, once you shatter that first limiting paradigm and trust the cell to police itself,
The pieces can be rearranged into something infinitely more powerful.
thanks for taking this deep dive with us. Keep questioning the paradigms in your own bioprocesses, and we will catch you on the next one.
You've been listening to Biomanufacturing and Fermentation Technology with Prasad Urnala. Thank you for joining me. Until next time, keep engineering biology into reliable manufacturing.